these particles are shrouded by a lot of mysteries which are yet to be resolved, and we need to know a lot about the nature, multiplication, and mechanism of replication, especially regarding these microorganisms. And to understand them precisely, we need to know a lot about them by applying varieties of techniques for studying them. And today we have seen that there are biophysical methods like electron microscopy, X-ray diffraction, etc., which gives us an idea about what kind of physical organization and what kind of chemical composition these entities have. Now, in addition to that, we have seen that there are sets of methods which are based upon the serological aspects. That is, either we need to find out the presence or absence of the viral specific antigen, or maybe we may check the presence or absence of antibodies produced inside the host directed against the infective viruses. So these kind of serological analysis largely have been discussed as hemagglutination assay, which is indirect estimation of the capability of viruses to see that how many of the RBCs or at what dilution the serum or the sample is able to cause agglutination of RBCs. So this is an indirect assay, an estimate of the number of viral particles present in the sample. Opposite to this, if we need to check the presence of antibodies directed against this hemagglutinin product or hemagglutinin type antigen, then we need to distribute varying concentration of the serum sample and to see that these RBC agglutinating antigen is inactivated at what concentration. So the highest dilution which gives the inhibition of hemagglutination reaction is referred to as titer. To make it simple, if a serially diluted samples up to 1:32 dilution is able to show hemagglutination, then 32 is regarded as a titer. So that is how we can conservatively carry out the estimation of the total quantity of the viral antigen or viral antibody products. Now, another class of methods that we studied today were based upon infectivity, where we try to grow and cultivate the viruses either over the laboratory animal or within the microbial culture cells or within the animal cell line or plant cells, whatever the host seems to be suitable to cultivate them. And we try to establish that the viruses have retained the infectivity, and by doing so we can also find out and approximate the number of viral particle within the sample. Now, another class of method that was also discussed were predominantly based upon the detection of very specific nucleic acid contained within the virus inside its core. And the most stubborn and powerful technique in this section seems to be the PCR, undoubtedly the most popular in all the molecular biology laboratories, and probably it has an answer to a lot of research challenges in the modern biology days. So using this PCR, we are able to amplify the specific sequences which are unique within these viruses by a way of PCR amplification, followed by its detection in the polyacrylamide gel electrophoresis analysis. And if virus-specific nucleic acid seems to be present, it indicates that these viruses are there. And also we are able to analyze the specific sequences and its importance within the virus or its relevance to its pathogenic capabilities. So many important and relevant answers can be obtained by a technique like PCR. We have seen a variant of qualitative and quantitative PCR, whereby we can also enumerate the total number of viral genome present in the sample. So it is also a way of accurately estimating the presence or absence of viruses. So in a way, there are many techniques which are regarded as classical or traditional methods, or maybe the modern one. But most of our present understanding over the viruses have been accumulated by a very ingenious but traditionally used techniques. Some of the basic information regarding the viruses, which laid the foundation for modern day research in biology and especially molecular biology, are based upon the foundation stone, which was in the form of work carried out by simple and classical analytical techniques used for viruses. And by way of these simple techniques, we have generated a lot of important information. And of course, now in the hands of molecular biologists, they have a much powerful and much ingenious techniques at their disposal, which are not only fast, but extremely precise, regulated, and very much target-oriented. And this is, in fact, not the end of the modern techniques. With the advancement of science and technology at a very rapid pace, we anticipated that many new, yet powerful, and yet effective techniques will be discovered, and the studying of viruses would become much simpler, much comprehensive in every aspects. Till then, we can see that the viruses are a potential threat to the human being, to animal, to plants, to bacteria, and all sorts of life. But at the same time, they are not only foe of the human being, they are friends of the human as well, because they are very significant and powerful tool in the hands of molecular biologists, whereby very useful and progressive genetic manipulation have been made feasible. So viruses is something which need to remain among us, but should remain within our control. And for that, we need to have better and better, more powerful, more elaborate techniques to study them from time to time. We have a list of references at the end of the presentation, and we also invite that the viewers can send an email to any of the speakers of today's session. We would be happy to respond to any queries from the student communities across the state, and we strongly recommend that some of the references which we have recommended at the end of this presentation are really important and worth reading. Additionally, the electronic resources available over the World Wide Web has a lots of useful information over the important subjects. Thank you all, and all the best.
SANDHAN, All Gujarat Integrated Classroom.
સેટેલાઇટના માધ્યમથી જોડતી કડી એટલે સંધાન.